So now that we have two pyruvate molecules from the original glucose molecule of glycolysis, we now need to prepare for further energy harvesting with our next step, the oxidation of pyruvate. This is sometimes called the transition step. In fact, it's my favorite description of this because it's the transition between glycolysis and the citric acid cycle. And it's really just one step process. This takes place directly outside of the mitochondrion and basically prepares the pyruvate to be made into a form that can be used by the mitochondrion. Please note that the transition step happens twice because there are two pyruvates from glycolysis. Also, don't forget that enzymes carry out each reaction and are not shown right here. The inputs of this reaction are two pyruvates, which came from glycolysis, two NAD pluses, which come from the electron transport chain, and then two coenzyme A's, which come from the citric acid cycle. First, pyruvate approaches the mitochondrion. I know you were expecting mighty. Well, I'm done with that. The third carbon group breaks off of the pyruvate as carbon dioxide, which is a waste product of cellular respiration. But because this bond is broken, a hydrogen ion and two electrons are free and reduces the NAD plus to NADH. This remaining molecule creates an acetyl group, shown here, which can now combine with coenzyme A to become acetyl coenzyme A. Now it is ready to enter the mitochondrion for the last process of energy harvesting, the citric acid cycle. So these are the outputs of the transition step. First are two acetyl coenzyme A's, which are now ready to go into the citric acid cycle of the mitochondrion. Two carbon dioxides, which are just expulsed as waste products and is also the source of why you exhale carbon dioxide. And then two NADHs, which will go to the electron transport chain, which will be the topic of our next video. But before we do that, we need to complete energy harvesting by heading into the citric acid cycle. So now that pyruvate has been converted to acetyl coenzyme A, it can now enter the mitochondrial matrix of the mitochondrion. Please note that the citric acid cycle is a repetitive reaction, and that the final product of the reaction becomes the initial product of the next cycle. Also, this reaction happens twice since two acetyl coenzyme A's are made from the previous step, and before that, two pyruvates, because again, we just split glucose earlier during glycolysis. The main function of the citric acid cycle is to complete the harvest of all of the hydrogen ions and electrons that we can get from that original glucose molecule that started in glycolysis. These are then sent to the electron transport chain to help make ATP. The citric acid cycle requires the following inputs: two acetyl coenzyme A's, which came from the oxidation of pyruvate or transition step. We need two ADPs and inorganic phosphates, which are used by the cell and brought here to be recharged by substrate level phosphorylation. We need two NAD pluses, which come from the electron transport chain. And then there's a new electron carrier called FAD. And FAD also comes from the electron transport chain to be reduced into FADH2 in one of the steps of the citric acid cycle. We also need water. Water helps to split up a lot of these molecules so we can have more access to hydrogen ions and electrons. So step one, acetyl coenzyme A enters the mitochondrial matrix. Coenzyme A is removed by the two carbon acetyl group via hydrolysis reaction. So we see water has entered the system, and what's going to happen is there will be an OH group on one side and a hydrogen on the other. And this allows for the splitting of the coenzyme A, which allows for oxaloacetate and the acetyl group to be converted into citrate. In step two, citrate is converted to isocitrate. Again, this is an isomer, meaning the same chemical formula, it's just slightly rearranged. But this simple rearrangement of the carbon groups is not shown here, but please know that this arrangement is necessary to carry out step three. So in step three, the 6-carbon isocitrate is then oxidized to 5-carbon alpha-ketoglutarate. This happens by removing a carbon group as carbon dioxide. As this happens, it frees another hydrogen ion and 2 electrons, which are then used to reduce NAD plus into NADH. NADH then delivers the electrons and hydrogen ions to the electron transport chain. In step 4, it's kind of a similar reaction. A 5-carbon alpha-ketoglutarate is then oxidized into a 4-carbon succinyl coenzyme A. This happens by removing a carbon group as carbon dioxide. As this happens, it frees another hydrogen ion and 2 electrons, which are then used to reduce NAD+ into NADH. NADH then delivers the electrons and hydrogen ions to the electron transport chain. Simultaneously, another coenzyme combines with this 4-carbon molecule, and it's shown by this blue molecule we see here. In step 5, the coenzyme A that is found on succinyl coenzyme A is replaced with a phosphate. Due to this, an ADP is attracted to the molecule and picks up the additional phosphate to make ATP. This is the source of substrate-level phosphorylation in the citric acid cycle. After that is removed, succinyl coenzyme A is converted into succinate. In step 6, succinate undergoes a dehydration reaction, which causes succinate to lose 2 hydrogen ions and 2 electrons to be picked up by a different electron acceptor called FAD to become FADH2. This takes the hydrogen ions and electrons to the electron transport chain to help make ATP. After this, it is converted into fumarate. In step 7, water is then added to fumarate to make malate. This addition of hydrogen ions and electrons allows for more energy harvesting to occur. In step 8, malate is converted into oxaloacetate, which frees up the last of the hydrogen ions and electrons needed to make ATP. It then becomes oxaloacetate, which can be used again to bind to the next coenzyme A and repeat the process again by making citrate. Now that the citric acid cycle is complete, the original glucose molecule has been stripped of any hydrogen ions and electrons that can now be used to make ATP in the electron transport chain. So here are the outputs of the citric acid cycle. First, 2 ATP are made and are going to be used by the cell. There's 6 NADHs and 2 FADH2s. And if you're wondering why the numbers seem off, it's because, remember, this cycle happens twice, and both of those go to the electron transport chain. We then create 4 carbon dioxides, which exit the cell, and in humans, we exhale it out of our lungs. In our next video, we'll be looking at the oxidative phosphorylation by the electron transport chain and see what the heck all of these hydrogen ions and electrons were being harvested for.